for photosynthesis. And higher plants have chlorophyll A and chlorophyll B as well. So they see that similarity. Green algae also, when they do photosynthesis, if they have extra sugar that they're not using right away, they'll store it in the form of starch. And that's what plants do. Terrestrial plants tend to do that as well. And terrestrial plants, when they store up their starch, they actually store it in their chloroplasts. So plastids in general. And then when you look at some green algae and you look at their cell walls, they have cellulose in the cell wall, hemicellulose, pectin, and those are the same ingredients that we were already talking about when we looked at the cell wall of plants. So there's other similarities between green algae and terrestrial plants, but those are three really strong points that make them think of that they're closely related and that the plants evolved from the green algae. So green algae is a super important group of algae. Yep. So they have mitochondria as well. Yep, you bet. So green algae are, did I put? I didn't put that in the, or did I? Oh, there it is. Phylum Chlorophyta or Chlorophyta. I'd like you to know the phylum name, please, of green algae. So phylum, depends who you talk to, that some people say Chlorophyta and some say Chlorophyta. They tend to say Chlorophyta, I guess. Okay, so phylum Chlorophyta are the green algae. Let me compare that. One other group that you worked with yesterday are the red algae, phylum Rhodophyta. Please know that too. Okay. So we're not going to get into all the algae. I'm just getting into some that are more commercially relevant and with applied aspects. So with red algae, when you look at them, again, there's pretty good diversity. They can be really small and you need a microscope to see them. So unicellular, they can be filamentous. They can look like seaweed-like morphology. And again, you might need a microscope or you could possibly see them with the naked eye as well. And in lab, you were playing around with Irish moss, which is not Irish moss. It's not a moss. It's actually red algae. And we were boiling it to extract the agar and the carrageenan from it, which is used as a thickening agent. And I understand that that happened toward the end of lab, and you put them in to cool down, I think, into the refrigerator. So we're saving those till next week so you can see, if you didn't get a chance to see how they thickened up. But agar and carrageenan, if you look at different store-bought products, you will see that showing up in like ice cream, for example. Carrageenan shows up in ice cream. You can eat agar as well, and it's referred to as agar. And so it's found in a lot of foodstuffs, cosmetic stuff. And so the red algae have it as part of their cell wall. So it's a strategy that they have. Agar and carrageenan, what it does is it allows their cell walls to be flexible and slippery. So, and then it kind of sloughs off over time as they make more. And so it can actually be a way for them to make sure that their body isn't covered by organisms. So they can slough off that over time just to kind of self-clean so they don't get covered and shaded out by other organisms. It can also kind of act as a sunscreen. It helps so that really intense sunlight isn't going to make it all the way through and start damaging parts of their body. So for them, they use it kind of as a protective mechanism in a couple of different ways. We use it in the food and cosmetic industry primarily for that thickening and creamy texture. And then some are very brittle or hard. They can actually have cell walls that have a lot of calcium carbonate. And so that makes it actually very rigid and brittle. And so the most famous example of that are the coralline algae that are on the coral reefs, and they just look like part of coral, but they're actually coralline algae. So you have red algae, again, quite a bit of diversity with this group too, with this phylum, and then some useful qualities. And again, just some visuals showing the different morphologies. And then on the bottom image is Irish moss, Chondrus crispus. Okay, I'll talk a little bit more about them in part 2. The last group I wanted to mention are the phylum Chromophyta, which are the diatoms. Okay, diatoms are, hopefully, did anyone look at the diatoms from fresh water? Did you see some in that sample? Okay, good. We had some leftover water from a while ago, but we thought, we better get fresh because it wasn't showing much. So the fresh water hopefully showed some beautiful diatoms. So you can see from this image, those are all diatoms. And again, they're quite diverse in their morphology. Most of them are unicellular. You definitely need a microscope. And they have silica in their cell walls. So they have a very rigid cell wall that doesn't decompose very readily. So it has a lot of applications. Beautiful to look at under the microscope. People have, I'll show you some pictures here where people will collect diatoms and then make art out of them because they're just so beautiful to look at. So this has been, an artist took all those diatoms and used a little bit of color addition to make them look that way. So this isn't color, this is natural. So this is red algae. All the red is red algae, and then gold color, those are diatoms that are attached to the red algae. I just thought that was a really beautiful picture. Every year they have competitions of microscope pictures, people looking at things under the microscope, and then they take pictures and enter them into competition. So this one, I don't know, it's called Small World Competition. I don't know if they won a prize with this one or not, but they entered. I thought that was really cool. But you can see that lots of different morphologies. This was color enhanced to show, but just beautiful, beautiful imagery, very complex. That's really close up using an electron microscope, just amazing detail. Here is an artistic rendition of many different diatoms all put together. Very creative. Another one. So certainly has sparked creativity with artists and making these different visual images. But we make use of them too. So let's see here. I'm going to pause.